Okay, so good evening, everyone. My name is Ankita. I am one of the TAs for this course of Basics of Biology by NPTEL. And this is week 4 of the live interaction session. And today we will basically be looking at three major topics. Or we'll focus on three things. The first is a recap of week, and finally, we will look at some sample problems for the upcoming assignment, which is for week 5. So, to actually go through the recap of this week, what we will do is we'll go through all of the presentations overall and try to brush up all the topics that were already discussed in extensive detail in the lecture videos, uh, and just revise the whole concept overall. So the overall theme of the entire course has been centered around understanding living organisms, as you are already quite aware. And first, we started with classification in week 1. Then we went into some understanding of evolution in week 2. Then we understood cells, the properties of cells in week 3. And finally, we are moving towards biomolecules in week 4. And we will take up the other topics in the upcoming weeks as well. So these are all different facets that altogether comprise or constitute the survival of a living organism. So when we talk about biomolecules, majorly there are 4 different types, as you're already aware. The first is the nucleic acid, which can either be DNA or RNA, and that is majorly concerned with carrying the genetic material of an individual or of an organism. And one of the key functions that is associated with the genetic material is that it has to be transferred from one generation to the next with absolute fidelity without compromising the information that was present in one generation. You have to make sure, not you, the organism or the system that is in place, that has to make sure that the information gets transmitted to the next generation with absolute fidelity and accuracy. The second type of biomolecule are lipids. Lipids are majorly concerned with energy production, and they are considered to be little building blocks. And these building blocks give rise to much larger entities or components within the cell. The third major type of biomolecules are carbohydrates, which are also building blocks that are involved in energy production, but they are composed of different smaller molecules and compared to the lipid moieties. And obviously, we have looked at how, and we will also go through it in today's interaction session. And finally, the fourth and one of the most important functional components of a cell are proteins. Majorly, these are the like all the information that is encoded or that is present within the genome of an organism, that is translated or that is brought to an organism's function by these building blocks. So these are proteins. So whatever information you have in the genome, that information needs to be converted to actual function. So if you have XYZ genes, those have to be translated or converted into XYZ proteins, and then those proteins will perform some function in some pathway that will allow the cell to sustain itself. It might be survival, might be in some other aspect, but it would have some of the other role to play in the cell's lifespan. And they are also often functional as enzymes, which are biological catalysts. And thirdly, we see that they have major roles to play in metabolism as well. So first, we are looking into nucleic acids in lecture 1. So like we have already studied that most organisms contain DNA as the genetic material, but only a few, for example, viruses, even within the entire population of viruses, only a small proportion of them contain RNA as the genetic material. And these are altogether the entire genetic content of the cell, that is DNA or that might be RNA. These are biopolymers. When we say polymers, it means that it is a larger molecule that is made up of smaller entities or smaller components, right? So those smaller components are monomers, and their nature in general is acidic. We'll understand why it is acidic. Now, eukaryotic cells, whether it be plant or animal, there the nucleic acid is present within the nucleus, but in prokaryotes, it's present majorly in the form of a nucleoid, as we have discussed in the previous week as well. And it is present in a free form, which is not really well separated from the rest of the cytosol. And first nucleic acid isolation was carried out by Friedrich Miescher in 1868. So this was actually the experiment carried out by Frederick Griffith, where he identified that this is the component of the cell that is actually acting as the genetic material. So for that, what he did was he took two different strains of Streptococcus pneumoniae, one which was virulent, which means which can cause disease, versus another which is denoted as S, and the other one is avirulent, or it is denoted as R, which is not capable of causing disease and will not result in the death of mice if injected into them. So so basically the entire experiment was divided into four parts. In the first part, he injected this S strain, which was alive. You can say it's a live bacteria, live S or virulent strain bacteria were injected into mice. And because these were live active bacteria, they caused disease and within these mice, and those mice could not survive. The second experiment was where he actually used heat. Now, because heat actually leads to denaturation or degradation of proteins, what happens is that the virus no longer is able to function. And under those circumstances, the virulent strain is dying, virulent bacteria is dying, and it is when injected into mice, it is not causing any disease and the mice are able to survive. In the third set, what he did was he mixed this non-virulent strain. In the third set, he actually did not mix. He injected the R strain, the avirulent strain, and obviously the mice did not die. But in the fourth part of the experiment, when he mixed these two, when he injected mice with non-virulent strain and the heat-kill virulent strain, he saw that these were actually able to die. So that's what they were able to understand, that the live bacteria and these heat-kill bacteria, when in combination, are capable of causing death in these mice. So that was one of the landmark experiments that actually brought forth the idea that yes, DNA might act, or nucleic acid might act as the genetic material within for most living organisms. Now we come to the structure or the arrangement or composition of a nucleic acid. So a nucleic acid, whether it be DNA or RNA, majorly has three components. The first is that it must have a nitrogenous base. When we say nitrogenous base, it means, if you look here, this is the base which is present with a base. Majorly, there are four bases, whether it be DNA or RNA. So there is adenine, guanine, thymine, cytosine, or there is adenine, guanine, uracil, and cytosine in case of RNA. So this is the nitrogenous base. There is one sugar. In this case, that is the ribose sugar. For RNA, it is just ribose, whereas for DNA, it lacks an OH group, and instead, in place of at the 2' position, in place of hydroxyl or OH group, there is just an H, which is why it gives the sugar is said to be a deoxyribose sugar, and such a deoxyribose sugar will combine with the nitrogenous base and phosphoric acid, otherwise known as a phosphate, PO4 2-, to produce or to form the entire nucleic acid. So this phosphate group that is present along the backbone. So if you look at the structure of a DNA, majorly its backbone, it's double-stranded, of course, but its backbone is made up of a phosphodiester bond, right? It has phosphate and it has esters in the backbone, and because of the presence of this phosphate, it confers a negative charge along the entire length of a nucleic acid or of DNA particularly. So that is where the backbone has a negative charge. But towards the interior, so if you have these two strands, these are negatively charged, but the space between these two, that is taken up by the nitrogenous bases, which are 9-membered rings majorly, and they are actually responsible for the huge diversity in nucleic acid sequences. And we can actually study that. So we'll get into details of it as well. So the phosphate backbone, like we said, the phosphoric acid forms the backbone. Then the sugars which are present, we have ribose and deoxyribose sugar. Then the nitrogenous bases, which are adenine, guanine, thymine, cytosine, and uracil in case of RNA. Those are also present here. But what is most important here, or rather one of the more important factors that need to be considered, that is between these two, the reason why this double-stranded nature of nucleic acids, or rather particularly of DNA, is actually of concern to us, or it's actually relevant to us, is because there is a complementarity that is obeyed within the double-stranded nature of DNA. So there we see that A is forming double bonds or two hydrogen bonds with T, while G is forming three hydrogen bonds with C. So why is there such a base pairing? We come to that later. We see that Chargaff's rules are also actually coming into consideration here, but we'll come to that later. So we are coming back to the nucleic acid structure. So there you can see that here we see that this is the nucleoside. When it has three phosphate groups, alpha, beta, and gamma, these three phosphates together form the NTP, which is the nucleoside triphosphate. So that is the entity or the unit that gets added to form the polynucleotide chain. So individual entities, they one by one, they keep on getting added and they form the polynucleotide. That's how it works. So during this addition process, what do you exactly need? Why is this addition occurring? That occurs because there is a 5' OH group and there's a 3' phosphate, which together form, what should I say? They actually are, there's a removal of water, and these two via a condensation reaction or ester formation reaction, they are forming this phosphodiester bond, and they're able to extend the polynucleotide chain. So effectively, one other thing it also does is that it renders a polarity to this particular molecule, and it runs in a 5' to 3' direction. So you'll have 5' at the 5' end, you will have 5' phosphate, and you will have a 3' OH at the other end. So that's how it is working. So we have established the polarity as well. So DNA is double-stranded, as you can see, and here you can see the bonds that are formed between these entities as well. So adenine, if you focus here, yeah. So adenine is forming 2 hydrogen bonds with thymine, whereas guanine is forming 1, 2, 3 hydrogen bonds with cytosine. So this is the base pairing which is formed. The question that comes is why. So one of the major reasons that are actually brought about is that so this actually is derived based on what is observed. You observe something in science, in any field of science, you observe something, and from that observation to analyze based on what information is known, and then you come to a conclusion, right? So we have 2 strands, and what is happening here is that based on the X-ray crystallographic data, which is one of these experimental methods of determining the structure for proteins and for nucleic acids as well, what actually suited the data, or what was in concordance with the observed data, showed that purine and purine, if 2 purines are forming hydrogen bonds with each other, then the entire groove is too wide. If it is one pyrimidine and another pyrimidine, it's too narrow. And if there's a purine and pyrimidine, then that is consistent with the X-ray data. It has the perfect match of the hydrogen donor acceptors as well. And therefore, 2 chains are effectively becoming complementary to each other. This is by accident a slide from the previous week's lecture. But coming back to complementarity, what does it exactly mean? It means that you can predict that if there is an A or an adenine at one side, then the corresponding or the complementary base which is present or which is pairing with that A will most certainly be T for DNA and uracil U for RNA. So both strands are effectively also running anti-parallel to each other, as you can see. So this is its 5' to 3', this is 3' to 5', and therefore they're anti-parallel to each other. Then comes Chargaff rules. So Shargov had explained that if you look at the entire base composition of DNA, you will find that purines and pyrimidines are always in equal amount. So the percentage A plus percentage G is always equal to the percentage C plus percentage T, which effectively means that number of A equals number of T, number of G equals number of C, and therefore you can predict. So while for different species, you might find a difference in this ratio of the bases, but if for one given species, it actually always remains constant, and you can use this property to actually identify the species and then use them in the process of classification, as we have previously discussed. And the sugar and nitrogen base and other phosphate components, they are always present in equal amounts or equal proportions. So that was still Shargov's rules. Then we can see on the right that the denaturation of DNA is something that's understood. So DNA, while it is extremely stable, it has to be denatured in vivo, which is within the cellular environment. And also if we are performing some experiments where that is a necessity, like for example, during PCR, you need to carry out denaturation. So mainly there are two ways to actually carry out DNA denaturation. One is via temperature. So subjecting it to very high temperature will result in the will break the hydrogen bonds. And the other way to go about it is to titrate it with an acid or an alkali. So during this process, this hydrogen bonding breaks, and that is effectively known as melting or denaturation. So when it is incubated at a low temperature, these two strands will initially, what happens when you have this denatured DNA, when you again bring them to which also happens during PCR, this lower temperature allows the hydrogen bond formation to again occur. And because of the complementarity, there is near perfect reassociation of the two strands via a process known as renaturation. So this renaturation kinetics are used to understand how complex DNA is, and obviously it has a role in PCR as well. So then we come to DNA denaturation and stability. If it is double-stranded, how are you denaturing it? You can use acid or alkali to actually carry out titration of the same. So when denatured DNA is kept at low temperature, you can see that this is the melting curve for DNA. But this is the temperature at which you have single-stranded DNA. Here you have double-stranded DNA, and here you have nearly equal volumes, which means that the temperature at which from the entire, so if you have say 100% double-stranded DNA and 0% single-stranded DNA, then the temperature at which half of the DNA has denatured, that temperature, so this is the dsDNA fraction, right? So when it is half, that temperature corresponds to the melting temperature of DNA. So that's how you can actually understand the kinetics as well. Coming to the process of isolation of genomic DNA. So we have talked about plasmid DNA isolation in the previous week's lecture. So when we are talking about genomic DNA, then first of all, the common step for every isolation protocol is that you have to lyse the cell first, right? When you need anything, any component of the cell which is inside, you need to lyse the cells first. So after lysing the cells with a detergent that contains a lysis buffer, we incubate them with proteinase K and SDS. What proteinase, as the name indicates, is an enzyme which degrades or which denatures proteins. So that's why you need to remove, when your objective is to get pure genomic DNA, you want to make sure that other cellular components are getting removed. So for that, we use proteinase K and SDS to release genomic DNA from the DNA protein complex. So isolation is next carried out using absolute alcohol, and then we can purify it using PC, which is phenol chloroform mixture as well. The aqueous and organic phases are separated, and phenol being a denaturant, it retains the proteins in the organic or in the organic phase. So finally, whatever you have isolated, the genomic DNA, that we can analyze on a 0.8% agarose gel, and a good proportion of genomic DNA gives an intact band, and like you can see here, you shouldn't get any visible smeared. Whatever extra we're getting, it's not really a smear, but here it's an excellent isolation of genomic DNA. Finally, I think in the last part, we are looking at the methods via which we can sequence DNA. So what you are doing is, you have cells, You have isolated, you have some sort of, say, mammalian cells or cancer cells, something like that. And from there, you have isolated the genomic DNA using the protocol that we just discussed. Now, after isolation, what you need, you suppose have tried to introduce a mutation, and now you need to verify, you need to confirm that yes, the mutation that you want to be incorporated has indeed gotten incorporated within that sequence. So for that, historically, there are two methods. One is the amplification with modified bases, which is Sanger sequencing, or the dideoxy method of DNA sequencing. And the second one is the Maxim-Gilbert method, where we use chemical modification and then cleavage. So these are the two techniques. First, we look at the dideoxy or chain termination of the Sanger method. Let's, I think, go to the figure directly. So there you can see that if you're starting with one particular set of or DNA sequences, what you're doing is first you are adding a primer, you're adding a radio-labeled ATP via which you're labeling the primer, right? Now you start PCR, but the PCR entire reaction mix has to be divided into 4 parts for 4 of these individual nucleotides. So in each reaction mix, you will have, apart from the enzyme and the primer and the template, you will have normal entities, which we have previously discussed, right? Nucleotide triphosphates. But in addition, and this is what makes this technique unique and such a brilliant move to actually figure out how you can sequence DNA. Here, Sanger proposed that you can also add a dideoxy NTP or a DDNTP, which means 2 of its OH groups are missing. As a result, when the 3' OH group is not available, further extension of that biopolymer or of that polynucleotide cannot occur, and therefore extension will get terminated then and there, because that is sort of an end termination or a chain termination method of sequencing. So what will happen is, when you within a reaction mix, you have ample number of DNTPs and DDNTPs, then it becomes a matter of chance that in one particular position, suppose if you have DDATP and DATP in one particular reaction set, then in n number out of n number of places, say n minus x places have incorporated the DNTP, and therefore reaction is proceeding fine. In the remaining places, DDNTP is incorporated and therefore reaction cannot proceed. So that you can actually figure out, and you can identify, oh, where exactly has the chain terminated, and you'll be able to know that, you'll be able to trace it back, that this particular position within my sequence of interest contains A or does not contain A, vice versa. So that's how the dideoxy method exactly works. And then we have the Maxim-Gilbert method, which is a completely different approach to go about it. And there we rely on chemical modifications and the follow-up cleavage. So here we have different sort of treatments. Yeah, here we can see. So for each, here are 4 different reaction sets are present here as well, but the deduction from these reaction sets is actually quite different compared to what we see in Sanger's method. Here, the deduction is for G, G plus A, T plus C, and C. And for that, the chemicals that are used are DMS or dimethyl sulfate, formic acid, hydrazine, and effectively we are able to see what is the change that is occurring, right? So after you have carried out individual reactions, we use piperidine, which is what will replace the modified base, and the next to the modified base, it will be able to generate a cut, and therefore the phosphorester bond would be cleaved. So after you have done that, and after you run it on a gel, you'll be able to actually reduce the sequence of the DNA or of the region of the genome that you were originally interested in. And finally, we come to RNA structure. So majorly we have, this is mRNA particularly, majorly we have a cap, a 5' and 3' UTR, a poly A sequence, and the coding sequence. Sorry, and the coding sequence. Now, RNA isolation is also, it's actually a very stringent protocol, because RNA is very prone to degradation by RNases, which are literally everywhere around you. So these have to be done with a lot, with a lot of cautiousness and with a lot of care, and therefore you will be able to get a good yield of RNA. So here we get the tissue and it is homogenized. Oligo-DT beads are used because, like you saw here, the RNA structure has poly A. So if you add a poly T containing bead, it will bind and it will use the weight of those beads to actually precipitate it upon centrifugation. Then we wash with the requisite buffers, elute, and collect. Right. So with this, we wrap up the first idea, and that's we talked about nucleic acids, DNA, RNA, their structure, their composition, some of their basic properties, the protocols for their isolation, and pretty much covered the part with nucleic acids. Next, we'll look at lecture 2. Just a second. Yeah, where we will actually delve into carbohydrates, and we'll just start that in a minute. Yeah. So beginning with the second lecture for this week. So when we talk about carbohydrates, they are mainly used as a source of energy, as we have described. And the major producers of carbs are obviously plants, because they're obviously carrying out photosynthesis, right? And they use CO2, H2O, and sunlight to do the same. Carbohydrates are also present in very simple form as monosaccharides, but they can also be very complicated, such as polysaccharides and glycoconjugates. So it's a huge class under which you'll have varying complexity within the molecules that are included within the same. Then here we're first talking about monosaccharides. So simplest carbohydrates can be aldehydes, ketones with 2 or more OH groups or hydroxyl groups. The backbone has 3 to 6 carbons with hydroxyl groups attached to it. We can see here multiple examples. There can be 3 carbon groups like glyceraldehyde, 4 carbon groups like erythrose, 5 carbon groups, most immediate example ribose, arabinose, etc., and of course the hexose sugars such as glucose, fructose, etc. Now, monosaccharides, the carbon atoms have in monosaccharides there are carbon atoms which have the chiral center, and this is the reaction between the first carbonyl and the fifth carbon group will give you cyclization or will give you the cyclic form of the same sugar, which is actually what you can see in the figure on the right as well. So this is the linearized or the chain version of Fischer projection of the glucose, and upon cyclization, when the first carbonyl and the fifth carbon, they are actually forming the cyclic form, you'll be able to get glucopyranose forms of the same sugar. Now, when we talk about monosaccharides, then majorly there are two forms of it. There is the hemiacetal and hemiketal format. So when there's a reaction between an aldehyde group and an alcohol, that is giving rise to hemiacetal, and that is forming the six-membered pyranose ring, much like what we just talked about. And here also you can see the two projections. One is the Fischer format, which is the elongated or the straight linearized representation of that molecule, whereas when we talk about Hayward projection, then it is forming the cyclized version of the same molecule. So that's what we see here, and here we can acknowledge that yes, that is hemiacetal formation via the interaction or the linkage between these two. Then comes hemiketal. In ketal, as the name indicates, there must be involvement of a keto group, which is the point of differentiation from hemiacetal. So a keto group and an alcohol are forming a linkage with each other, ketal linkage with each other, and the resultant structure that is formed in this case, compared to, in that case, you had aldehyde where there was one more carbon, versus when you talk about a ketal or hemiketal in this particular case, then you have like in case of hemiketal, you will have the five-membered furanose ring. Like there's one less carbon, so it's a five-membered ring, and it is a furanose, like fructofuranose ring would be formed in this case. Now, the cyclization is generating an additional chiral center and is contributing. Now comes a very important terminology, which is the optical activity. So for any sugar molecule, any carbohydrate or particularly monosaccharide molecule, if the sugar is able to rotate the plane of polarized light. So plane polarized light, when is incident upon the sugar, if it is able to change the direction of that light, then it is said to be optically active. Now this activity or this rotation can be either towards right or towards left. If it is towards the right, it is known as dextro-rotatory, indicated by plus sign or the D sign, versus levorotatory, where it rotates the light, the incident light towards the left, then you call it as levo-rotation, and it is denoted by the L sign or by the minus sign for that particular sugar. Then come enantiomers. Enantiomers, if we just simply put it, the most common definition that we've been learning for a while now is that they are non-superimposable mirror images. Here it's just mentioned as mirror images. So if you imagine that there was a sort of mirror kept here, then the two sugars would have points lying at pretty much the same distance from the mirror plane, and they would form a different molecule. And these two, if we just directly superimpose one on top of the other, they are not superimposable, and therefore these are known as enantiomers. So that's what we get, L-glucose and D-glucose. So the L and D forms are looked at here. So these sugars are compounds where the last asymmetric carbon is present in the same orientation. So for that, let's look at some examples. So previously we also talked about L and D, but that is with respect to optical activity, and it was denoted by small l and small d. So that's for levorotatory and dextrorotatory. But when it is represented in capitals, that is actually a different form of classification. And there you can see that this is the last carbon and there's the second last. So in this case, you can see in both cases, OH is lying in the same direction, or it is both the OH or hydroxyl groups are oriented in the same direction, then it is represented as D-glucose, versus when they are in the opposite direction, you would call that as L-glucose. So that's another way of classifying these sugar molecules. So that's what we see here. And next we come to, so we are talking about we will like it's already been discussed, but we'll be looking at several different ways of classifying sugars, particularly monosaccharides. The first was with respect to optical activities, levo and extraregulatory, then the L and D forms. Now we are looking at epimers. So epimer, the definition for that are isomers of the same sugar molecule, but where they have a difference in the OH and H configuration of the second, third, fourth atoms of glucose. For example, here if you see that if you look at just the formula of the compounds, it's the same, right? It's just that how they are linked to the carbon when you are carrying out Fischer, or when you are trying to demonstrate the Fischer projection, then you see OH and H in the second, for the second carbon, are oriented differently in these compounds. So these are epimers of each other. So that's the differentiating factor between these molecules. Then come anomers, anomeric carbon. It can have alpha or beta form. So they differ in OH group configuration of the carbonyl group carbon and anomeric carbon. So there you can either have rotation towards the right or towards the left. If it is towards the right, it's the alpha form. If it is towards the left, it's the beta form. Next, we are looking at mutarotation, yet another property. So mutarotation actually looks at monosaccharides that contain asymmetric carbon atom and that are able to rotate plane polarized light. So this rotation, particularly when you talk about glucose, it's with respect to the fact that if you just let glucose stand in solution, you will see that there is a change in optical rotation in such a case or under such circumstances. So we have alpha D glucose and we have beta D glucose. Then come disaccharides. So we were talking about monosaccharides all this while. Disaccharides are when you have these, they are formed when you have these individual monosaccharides that are that utilize their terminal OH group to form a glycosidic linkage to form disaccharides by losing one water molecule. So when you actually add water, it can be again hydrolyzed to form individual monosaccharides. So different monomers can participate to give you different disaccharides sugars. So that's what we are seeing here as well. So there's hydrolysis or there can be condensation to actually result in the reversible reaction between the two. Now, the linkage that is formed in this case is a glycosidic bond or a glycosidic linkage. So when you have OH group on the anomeric carbon of a sugar, and when that reacts with an alcohol or it reacts with another sugar, that's an O glycosidic linkage. So you have OH group. The OH group is of course required for any glycosidic linkage, but the other participant in such a case is an alcohol. Compare that to N glycosidic linkage by the OH group. Just one second. Where the OH group is, it's reacting with the anomeric carbon of a sugar. When it is reacting with the amine group, and then it is resulting. So for amine, it's denoted by N, but for the O glycosidic linkage, since it is reacting with an alcohol, it's denoted by O. So they are present in nucleotides, RNA, and DNA. Then come the alpha and beta bond for the glycosidic linkages. So here you see maltose and lactose, which are two disaccharides. And you can see that the linkage between them is actually different. You can see that when the linkage is direct between 1 and 4, if the anomeric hydroxyl group, the OH group basically, if it is below the hydrogen, so OH group is below the hydrogen, then that is the alpha bond and it is denoted in this manner in the Hayward projection, and that is giving rise to an alpha bond or alpha glycosidic linkage between the two. Compare that to the beta bond where the anomeric hydrogen hydroxyl is above the hydrogen, and then you can see that it's a zigzag photosynthesis Z scheme. If you remember, it's much like that. It has a zigzag orientation and it is giving rise to a beta bond in the formation of the disaccharides. Coming to polysaccharides, when different monosaccharides or disaccharides, they condense, they effectively give rise to polysaccharides. They are the storage form, they are the format in which sugar is stored, and then it can get hydrolyzed to give rise to monosaccharides that can actually have some involvement in metabolism to reducing equivalents, and ultimately it gives us energy. So the terminal sugar in case of disaccharides or polysaccharides has a free anomeric carbon and it's called as the reducing end. So that's what we talked about polysaccharides. Now, sorry, it's the same slide. Then how do you actually know that they are reducing sugars? If this has been explained by Sir, you use Benedict's test or you use Spelling's test and you'll be able to see that they are reducing sugars, right? So that completes lecture 2. Coming to lecture 3, just a second. This is actually a compiled document with both lecture 3 and 4, a compiled PDF with both of the lectures. So first, we are going to talk about cellular metabolism overall. Now, metabolism very broadly can be classified into anabolism or catabolism. Catabolism is where you cut, which means where you break down, right? So you are breaking down some bigger molecule, producing smaller molecules, and as a consequence, you are generating energy. Compare that to anabolism, where you are bringing things together or combining things together to produce a bigger molecule, and that's a biosynthetic pathway. You're synthesizing something, and that process is known as anabolism. So for glycolysis, it's actually central to carbohydrate metabolism. It's the universal pathway found in prokaryotic or eukaryotic cell. Glycolysis is one of the most fundamental processes that are operational in overall cells, even in cancer cells. It's a major source of energy. So basically what it does, if you just break down the name, if you do catabolism for the name, you'll see there is glyco and lysis. Glyco in particular here is glucose. You have a 6-membered sugar, and that is broken down into 2 3-membered sugar molecules that can then go forward for producing energy. Now, after this three carbon sugar is produced, which is pyruvate in this case, it can proceed towards either direction based on what is the oxygen availability within the cell. So if there is sufficient oxygen, it will proceed towards Krebs cycle, or if it is in more majorly an anaerobic condition, it would proceed towards anaerobic oxidation. And therefore, it's very important for a cell to actually have an operational and functional glycolysis to cope with different type of stressful situations, particularly with respect to oxygen. So it's a 10-step chemical reaction. Sorry. It's a 10-step chemical reaction, and it involves a lot of different enzymes that ultimately help in use up or that ultimately use up glucose to produce the downstream molecules. So these are the different steps. I'll not go through all of them because it has been elaborated in a huge amount of detail. So you see it how it goes from glucose to glucose 6-phosphate. We can actually go through the steps here. So step 1 is that glucose phosphorylation. Then coming to step 2, which is conversion of glucose 6-phosphate to fructose 6-phosphate. Then comes glycolysis, which is in step 3 of glycolysis, which is from fructose 6-phosphate to fructose 1,6-bisphosphate. Then comes DHAP, which is it actually involves the splitting or the cleavage into GAP and HAP, which is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Then the 5th step is the interconversion or reversible conversion between these 2 molecules, but among them only GAP or glyceraldehyde 3-phosphate is able to proceed further. And from G3P, we actually get 1,3-bisphosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, phosphoenol pyruvate, and pyruvate. Right, so these are all of the different steps, and if you look carefully, you will be able to see that initially there is an investment of energy, which is that it requires ATP. So a cell which does it's never that a cell can have no ATP molecules, right? There is always a basal amount of energy which is present in the cell that has to be maintained, and that's crucial for survival. Now, with respect to that base, you can either go up and down, and based on that, you decide the energy status of the cell, that compared to the amount of ATP present, what is the proportion of ATP, and therefore you decide that how rich is that cell at that particular moment. So that is actually the energy status of the cell. So we see that after an initial investment of 2 ATP molecules, effectively, we then get 2 ATP molecules that are produced during one glycolysis step, and also one NADH plus H plus molecule is produced. So let me just come to this. Yeah. So this is the ATP balance sheet, which we can see. So during steps 1 to 4, you can see that 2 ATP molecules are required and they're invested, following which we are able to see that each NADH is equivalent to 3 ATPs. Now we can find that there are two NADH molecules that are produced for each glucose. So you started from glucose, you got 2 3 carbon molecules, right? You got DHAP and GAP. So you'll have 2 GAP molecules from one glucose, and therefore the entire energy currency, it actually gets doubled, and you'll have 6 in this case. Just give me a second. Yeah, sorry for the interruption. I was getting a call. So there we have, after investing 2 ATP molecules, we are getting 6 NADH molecules from one glucose, and there are 4 ATP molecules that are generated. So effectively, what you're getting after each cycle of glycolysis is that you're producing 8 ATP molecules, right? Now, the question comes, how is it at all getting regulated? So the reason why it has to be regulated is that it's not always necessary to perform glycolysis, right? So the reason for that, if you just follow this stepwise, you will see that the first step is that insulin, the hormone that controls the blood sugar, that is one of the key players, because glycolysis, the entire process is dependent upon glucose, right? So blood glucose is actually under the control of the steroid hormone insulin. So not steroid hormone, peptide hormone insulin. So insulin binds to its receptor in the cell membrane. This is its membrane receptor to which this hormone, represented by the white block here, it binds. After binding, what happens is that the receptor, which is now so actually insulin receptor is a receptor tyrosine kinase. It follows a particular pathway via which it is responsible for recruiting glucose transporters from the inside of the cell towards the cell membrane. So insulin has bound. What it is doing is it is recruiting glucose transporters to the cell membrane. What these are doing is that they are actually executing higher uptake of glucose into the cell, because the hormone is signaling that, okay, you need to keep things in check. The overall blood glucose is very high and we don't need it, and therefore you need to take glucose back into the cell and not allow it to circulate. So that's what happens, and it basically sends up glucose transporters to the cell, that you now carry out this transport and you bring glucose towards the cell interior and don't let it circulate. And effectively what happens is that the blood glucose would decrease. So when insulin levels would again further decrease, now when that is again regulated, you cannot continuously have insulin suppressing or controlling the level of glucose in the blood. What it does is, when insulin levels will decrease, the glucose transporter moves away from the cell membrane into the intracellular storage pool, and there it is recycled, and then vesicles are able to fuse with the exosome. So that's how glycolysis is controlled. And that's also, there are other controls as well, which sir has also discussed in details about the covalent modification of pyruvate kinase, where the dephosphorylated form is more active, and upon phosphorylation it's less active. And also there is allosteric regulation, which means that if you consider this as the enzyme, there is some shift in its 3D structure or its shape, as a result of which there is this variation. So that's what we see here. And then we have Krebs cycle, which is actually, so we had initially talked about how the presence of energy, or not energy, oxygen, how the oxygen availability within a cell will decide whether it will go for Krebs cycle or not. This is also, as you have studied, it's a very detailed and a very critical process within a cell, which actually is ultimately, not ultimately, it's one of the major stepping stones for energy production within the cell. So after glycolysis, if there is adequate oxygen supply in the cellular environment, then the cell proceeds towards the tricarboxylic acid cycle or the Krebs cycle. So it involves pyruvate, and it goes through these a number of steps where you can see so many cofactors involved as well. It goes through oxaloacetate, citrate, cisaconate, isocitrate, oxalosuccinate, alpha ketoglutarate, one of the major very important intermediates of Krebs cycle, succinyl coenzyme A, succinate, fumarate, malate, and again it cycles back to oxaloacetate. So that's the different one of the differentiating points from glycolysis as well, is that it's a cyclic process. So if you look at oxaloacetate, that's where you start when it combines with pyruvate, and it again comes back to OA again. So it's a cyclic process that continues, giving the name Krebs cycle, discovered by Hans Krebs. So if you look at the energy equivalents which are involved in this particular process, then we'll see that we have acetyl coenzyme A, we have alpha ketoglutarate, and all of the other intermediates. I'm not going through all of it. What you ultimately get is that after glycolysis, because you had 2 3-carbon molecules, which are pyruvates, 2 pyruvate molecules, each pyruvate is producing 15 ATP, and therefore total 30 ATP is generated. Then come the controlling steps or deregulating or crucial steps of Krebs cycle. There are 4 such rate limiting steps. So we have the first step, which is the conversion of pyruvate to acetyl coenzyme A as one of the major steps. Second, the first reaction, which is these are known as committing steps. So at every point, the cell has the choice to make whether it is going towards direction A or pathway A or pathway B. The cell actually makes that choice. So when it is doing that, it actually has to commit to a particular pathway, and one of the commitment steps is actually this. So it goes via when it is actually entering the Krebs cycle and citrate is synthesized, it cannot actually go back, and it cannot decide that, oh no, I don't want to go for Krebs cycle. So this is a committing step, and therefore it's one of the most crucial steps that actually initiate the process. So that's what we are talking about. Then there is isocitrate dehydrogenase that gets inhibited by ATP and NADH, whereas ADP and calcium are 2 entities that actually stimulate the same. You can see these here as well. And alpha ketoglutarate dehydrogenase gets inhibited by succinyl coenzyme A and NADH, whereas calcium stimulates those processes. So these are 4 very crucial controlling or regulatory steps that are involved in Krebs cycle. Then how it exactly regulates metabolism, it's also pretty much the same. I'm not getting into the detail. And its role in, it might sound crazy, but it is one of the reasons how we can actually support the hypothesis that organisms have been able, that as the earth evolved, like we initially discussed during evolution, right? That with time, how organisms have changed, how they have evolved, is how it depends on how Earth has evolved, right? So that evolution has actually, or that hypothesis of how the Earth has evolved is hugely supported by the Krebs cycle, because you don't find it in primitive organisms, but in higher, more complicated organisms that have adapted to this sort of environment, you need Krebs cycle to be present, and you need it to operate for those organisms to be able to survive. So that's what we see here. And next we will look at the final presentation for this topic, and then we can shift towards questions, and that would conclude our session. So here basically we looked at nucleic acids, we looked at carbohydrates in great detail, we saw the cellular processes that the carbohydrates were involved in also in significant detail, and now we are coming to lipids. So lipids are not, you cannot very generically define what a lipid is, because they have a large number of subgroups. So that includes fats, oils. You can see here there are fats, oils, steroids, waxes, and other related compounds. And majorly the similarity lies in how it lies actually in their physical properties instead of their chemical properties. So what you can actually say is that they are not, or you can say they are relatively not very soluble in water, but they are quite soluble in nonpolar solvents like ether and chloroform. So that's what we see. Then we are talking about one of the major groups under, or one of the major components of the entire class of lipids is glycerol, commonly called glycerine. It is a trihydric alcohol, which means it has three hydroxyl groups. So they can be obtained from diet, from fat, like from lipolysis or breakdown of fat, from glycolysis, which we have also seen. And it's used as a solvent. And in fact, nitroglycerine, you might actually find this as one of your assignment questions as well. So nitroglycerine is often used as a vasodilator. So it's a very commonly used medicine used for a lot of cardiac patients, and it's requisite for maintaining their normal blood pressure. So nitroglycerine is one such important component. So that's how you understand why studying each of these components, why at all studying biology is relevant, right? Because you never know what might have some application in treatment or in survival of an individual. So the more holistic your knowledge, the better. So that's what we see. Next come fatty acids. So fatty acids are aliphatic carboxylic acids. They have this common general formula, which you can see. Mainly they occur as esters in natural fats and oils, but sometimes they can also occur as free fatty acids, FFA, which is how you actually find them in blood plasma when they are being transported. Fatty acids that occur in natural fats, they're usually like straight chain derivatives, and they have an even number of carbon atoms, and that can either be saturated or unsaturated. When we say saturated, it is just made up of single bonds. There are no multiple bonds are not present, and otherwise it can also be unsaturated. Unsaturation means it has some, at some positions along the entire length of that particular fatty acid, you will find some double or triple bonds present. So according to this nature, they can be saturated, unsaturated, they can be branched chain fatty acids where you have one main chain and then it splits. You can have it in that way, or you can have fatty acids with some sort of substitution present within them. So saturated ones do not have them, which is something we've already discussed. And here it's very light, but you can see some examples of saturated fatty acids. I hope it's visible, like acetic acid, butyric acid, caproic acid, caprylic, capric, lauric, myristic. And what actually matters is the number of carbons that you see here and its more systemic name. So you have decanoic acid is capric acid, dodecanoic, which means it has 12 carbons, is lauric acid. Then we have docosanoic acid is behenic acid, which is 22 carbon, and so on and so forth. I particularly mentioned some of those because they are a part of your assignment. So that's how you need to make sure that you're studying these notes in appropriate detail, right? So then we come to unsaturated fatty acids, and so unsaturation basically means it has a double bond, right? Now that can either be a single double bond, or not double bond, it has to be or it should technically be a multiple bond. That can either be one multiple bond or it can be several such bonds. So monounsaturated or polyunsaturated, as we were previously discussing. And how these double bonds are present, or how other carbon groups are actually attached to them, that determines whether they are in cis or in trans. So they are separated, the double bonds are separated by at least one methylene group. The systemic name for a fatty acid is derived from the name of its parent hydrocarbon, and it has oic in its name as well. So we have octadecanoic acid, and the original parent hydrocarbon is octadecane. C18 is octadecanoic acid with two double bonds, octadeca dienoic acid if it has two double bonds. If it has three, it is trienoic acid, and so on and so forth. Then we talked about the classification of lipids. They can be simple, compound, or derived, and we get into details about them as well. So simple lipids are esters of fatty acids with several alcohols, we have fats, and we have waxes as well. Fats in the liquid state are oils. Waxes are esters of fatty acids with high molecular weight monohydrate alcohols. So that's what we see here. Then we come to triglycerides. So triacyl glycerols are esters of trihydric alcohol, glycerol, and fatty acids. So yeah, triglycerides, I'm not getting into all of these details. They are majorly, they're the storage, they're the energy storage packets. You can think about them that way. They are major lipids in the body, and in diet they have about 60, or they provide about 60% of the body's resting energy needs, very in a very compact area. They are necessary for insulation and protection. They are carriers of fat soluble compounds, and they give about the sensory quality of flavor and texture to the food that you talk about, which is not entirely desirable, which is why with age you have to actually keep your triglycerides in check, and that's what people do. It's not even about age, it's for just overall health and wellness. So that was the point. Then coming to waxes. So they are esters of higher fatty acids with higher monohydroxy aliphatic alcohols. They are long chain, straight chains of 60 to 100 carbon atoms. So they're very long chain carbons. As you know, they're involved in cosmetics, candles, polishes, etc. And they are forming the outermost layer for many organisms. Like it's a defensive strategy for many insects, many plants, that they have a waxy cuticle or waxy coating at the top of their body. And like we know, for example, for plants, to reduce the water loss by transpiration, there is often a waxy cuticle that's present. Those were the simple lipids. Coming to compound lipids, we see that they have other groups. Apart from, you have an alcohol, you have a fatty acid, but you also have some other groups. So those are compound lipids, and among them we have phospholipids, glycolipids, and other complex lipids. Among phospholipids, we're coming to phospholipids first. Let me just zoom in a little bit here. A bit too much. Sorry. Yeah. So phospholipids can majorly be classified into glycerophospholipids or sphingophospholipids, right? Here you'll find examples of each of these. Glycerophospholipids with several different X groups or head groups of constituents such as ethanolamine, serine, phosphatidylserine, phosphatidylcholine, phosphatidylethanolamine. They all have different functions. Among them, one of the most important which has also been mentioned, is the phosphatidylserine. It has a very major role to play, which we'll discuss in the assignment as well. And sphingolipids have sphingol or sphingo as the alcohol group, and what it produces effectively is sphingomyelin. So sphingomyelin, a backbone is sphingosine. It's a long chain fatty acid attached to the amino group of sphingosine, and what it effectively forms is ceramide. So the alcohol group at carbon 1 of sphingosine is esterified to phosphatidylcholine, producing sphingomyelin. Sphingomyelin is an important component of the nerve fibers. It forms that myelin sheath, which we are familiar with. Then these are their very different, very diverse groups of functions, and each of these functions are actually important. One of them is also part of the assignment, but yeah, I'm not going through each and every one of them. But coming to the next part, which are glycolipids. They differ from sphingomyelins because they do not contain phosphoric acid, and the polar head group function is actually provided by monosaccharide or oligosaccharides. The carbohydrate moieties that are present, they are also capable of determining what is the type of the glycosphingolipids. They can be neutral or acidic. Majorly, they are found in the outer leaflet, where they contribute to cell surface carbohydrates. They can act as surface receptors, blood group antigens, GM1 for pulverotoxin, etc. Then comes sulfolipids. They are cerebrosides, which contain sulfated galactosyl residues. They are negatively charged. Whenever you have sulfate group involved, the charge is there, predominantly in nervous tissue and in kidney. And when not degraded, or when failed to degrade, then it causes accumulation in nervous tissues. So lipid storage disorders are actually quite plenty. These are also for direct or recollection or direct memorization. So we have Tay-Sachs disease, Fabry's disease, metachromatic leukodystrophy, Krabbe's disease, Gaucher's disease, and so many more. And finally, we come to derived lipids, where we have cholesterol. Here only one example has been taken up, cholesterol, which can either be in its free or its esterized format. And the hydroxyl group on position 3 is esterified with a long chain fatty acid. LDL, HDL are two major different types, and it has been discussed in details, particularly a very important component of the nervous tissue. It forms a major constituent of plasma membrane and plasma lipoproteins. It's synthesized from acetyl coenzyme A. It's the precursor of all other steroids in the body, like all the sex hormones, corticosteroids. They are all cholesterol derivative. And also, unfortunately, it's a component of gallstones as well, and its chief role is a factor in atherosclerosis, causing several types of diseases. So with this, we are more or less done with the recollection of, or the recap of week 4. We will start the assignment and in just 2 minutes. Okay. Okay, so again, sorry for the interruption. I just needed a break because I was speaking nonstop. So we have already done the first part, which is a recap of week 4. We will now just go through the assignment, discuss it. You already have the answers for it. So the first question for this week's assignment was which of the following is a combination of enantiomers? Now, as we have already discussed that, enantiomers are non-superimposable mirror images. So if you look here, I've also indicated the answer right next to it, which is L-glucose and D-glucose. You will be able to see that if you look at the structures of these two, they are not, they are mirror images of each other, but they're not superimposable. So considering this as the mirror, you can see that each of the attached substituents to each carbon group, they are present at exactly the same location but opposite to each other, right? So that is the answer. And of course, if you look at the other options, they do not satisfy the same. You will find that all of the other options have both the sugars in the same orientation, which means that they are all D sugars, right? So that's why they do not actually satisfy this concept. Then question 2 was about glycolysis. And here we were asked that which of the following enzymes helps in the conversion of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. So we saw that this was the breakdown step, right? So you started with a 6-carbon sugar, which is glucose, and from there you're coming down to 2 3-carbon sugars. It's actually splitting the initial sugar into two, and it is producing DHAP and GAP, which are actually seen to be interconvertible, and the enzyme that operates in that step is actually playing a very crucial step, because that actually is deciding how much of the sugars will actually proceed forward towards glycolysis, because DHAP does not go with it, or does not go for further processing or conversion into pyruvate, but instead it has to be the equilibrium has to be shifted towards glyceraldehyde 3-phosphate to actually regulate the same. So we see that for phosphotriose isomerase or triose phosphate isomerase. That's the more common way of actually structuring that name. So that's what we see. In question number 3, we are being asked that the DNA or RNA are acidic in nature because of which of the following? So here we have the options as nitrogen base, sugar molecule, phosphate group, and both nitrogen and sugar. So if you remember, as we have discussed like just a few minutes ago about the entire composition of the nucleic acid and what are the different components, how are they present, how are they arranged, you will see that the acidic nature actually comes from the phosphate group, right? Because the other molecules, the nitrogen base, as you can see, it's basic, or it's not necessarily the nature, it does not have any acidic groups that, or it does not have any groups that can confer acidic nature to the entire molecule. And the sugar is of course also uncharged, and it does not do the same. So it is mainly the phosphate group which is responsible for contributing towards this acidic nature. So that's the question number 3. In okay, don't get intimidated by the figure, but yeah, this is another representation of the Krebs cycle or of the citric acid cycle. It's very detailed, but yes, it's just another representation to familiarize people with. So the question that was asked was about which of the following are decarboxylating enzymes in Krebs cycle. So for this, you simply need to recall, or um, you need to remember the Krebs cycle, which is a challenge, but it's very doable. You just need a mnemonic where you can actually remember the first letter or of each of the intermediates, and you can form that mnemonic as per your comfort. One of my friends actually does this, and it's easy to recall it because there are so many intermediates, it can often get confusing. But one very advantageous part, you can say, is that the citric acid cycle has been it has been written, or not written, it has, the nomenclature for it has been done in such a way that if you just remember the intermediates, you'll more or less be able to recall the enzyme, because you'll know which step that particular enzyme is working in, and that name is actually derived from the same as well. So that's an easy way to proceed with it. And here, since the question is directly asking us about decarboxylation, that is mainly done by isocitrate dehydrogenase. So here you can see CO2 is getting removed or released, and that's isocitrate dehydrogenase is the enzyme working or operating in that step. And the next step also we see a CO2 which is released, and that is done by dehydrogenase, alpha ketoglutarate dehydrogenase. You do not see any of the other steps doing this. And yeah, here we can see pyruvate dehydrogenase, but that's not a part of the options, and it's actually not a part of the Krebs cycle as well. So in case get a question where there is pyruvate dehydrogenase also is present as an option, you need to make sure that acetyl coenzyme A, after acetyl coenzyme A is actually formed, that is entering the Krebs cycle, not before it. So it's not necessarily a part here. The options are relatively more direct, I would say, but many times you might find a question where you might be confused between the two. So you can try to avoid that. Then we come to question 5, where unfortunately, this representation does not show TPP, but yeah, TPP or thiamine pyrophosphate, that is, that's a, you can call it a cofactor. It is involved in which step of Krebs cycle is the question we are being asked. So while this representation does not have it, if you go back to the representation that was provided in the original presentation or the lecture notes, you will see that it's involved in the first step, this decarboxylation step that I was talking about with pyruvate dehydrogenase. PPP is a cofactor for this step. So it's involved in this very crucial step of conversion to acetyl coenzyme, and it's involved in decarboxylation of pyruvate, option D. Coming to question 6. So you see many of the questions from the assignment of a Krebs cycle. I mean, if you weren't convinced till now, this should convince you that this cycle is so relevant and so crucial. And you, even if you cannot recall every step, its concept, its necessity, the intermediates that are involved, how it is fine-tuned, all of that, um, that's relevant in our case at least. So amino acids such as glutamine, proline, and arginine are formed using dash intermediates from the Krebs cycle. This is also a direct question, I believe. So you'll be able to see that alpha ketoglutarate, which is produced here, that is actually the step from where we get all of these different amino acids. So again, very, very crucial pathway involved in cellular metabolism. Coming to the next question, which of the following molecules has the maximum number of carbons in its structure? So if you remember, there was a tabular format where all of this detail had been provided, but just so that you can recall it, you have arachidic acid, stearic acid, palmitic acid, and myristic acid. You'll get the entire table, and you'll have individual number of carbons which are present in it. But here, just for reference and for your own benefit and recollection, you can see that this is the structure of arachidic acid. And if you look at the formula, the chemical formula for it, or the number of carbons present here, you'll see that there are 20 carbons. And among these options, this has maximum number of carbons. And that is the part that's most relevant to us. Then in question 8, we have glycerol. This is a pretty straightforward question because actually glycerol is serving as the backbone for so many of the lipids that we just talked about a while back. So glycerol is 2, 3, 4, or 5 carbon molecule. Then obviously most certainly it's a 3 carbon molecule that we have talked about. And the glycerol backbone actually distinguishes the glycolipids from the phospholipids, as we have discussed as well. Then if you remember that nitroglycerin is used as an anti-cancer drug, antihistamine, vasodilator, or a lubricant, then you remember that it's used indeed as a vasodilator, which basically means it's involved in dilation of blood vessels, which means that it increases the area through which, the cross section through which blood can flow, and it's often a treatment strategy used for many patients who have some deposition, atherosclerosis or some sort of deposition along their cardiac vessels, along their blood vessels, which results in increased pressure, and like blood has to be pushed with more pressure through those vessels, and therefore that's the distinguishing portion, I can say. So nitroglycerine is used as a vasodilator. Then in question 10, we were talking about, or we are directly asked, what is an example of docosahexaenoic acid? So if you remember, while I was recapitulating what has been discussed in that week, there we went through that table, and there we saw that behenic acid is an example of docosahexaenoic acid. It has 22 carbons, which you can see from the figure on the right. It's immense. It's a huge, really long carbon chain, and it's an unsaturated carbon as well. It's a saturated structure as well. So that's behenic acid on the right. You do not see any double bonds or triple bonds present here. So it does not have any unsaturation. Then yeah, in question 11, this is actually very interesting attribute, which is that which of the following phospholipids is related to the removal of apoptotic cells by phagocytes. So we had discussed a little bit about apoptosis, necrosis, and necroptosis, all of these processes, even about autophagy and how the lysosome is involved in it in our previous discussions. But the entire process of apoptosis, it has like multiple stepping stones or multiple how should I phrase it, approaches which all converge to the decision that whether this cell should live, or does it, does not, is its life not worth spending all of this energy on? So it sounds grim, but that's how the cell decides that if considering the availability of nutrients, whether or not the cell can survive. That decision is actually crucial, and it is made by the cell via multiple areas or multiple levels of control. So among them, one of the major apoptotic signals is phosphatidylserine. So what happens is, under normal circumstances within a healthy cell, phosphatidylserine is localized towards the inner leaflet of the plasma membrane. So if you remember, it's a phospholipid bilayer, right? So if you consider this side to be the cell exterior, this side to be the cell interior, then this is the outer leaflet and the inner leaflet, right? So I'm saying leaflet because it's a bilayer. So among these two layers, one forms the outer, one forms the inner. That's how the terminology is used. So phosphatidylserine in normal cells under normal circumstances is localized in the inner leaflet of the plasma membrane, that is, it faces the cell interior. However, when a cell has, when it has seen that it does not have the resources or the conditions to actually survive, what it then does or decides is that it will go towards apoptosis, and then this phosphatidylserine is shifted towards the outer leaflet, and that serves as an eat me signal. If you go through books, that's actually the terminology used, which means that the cell is saying that I cannot survive, and it's calling for other cells. Here it's mentioned as phagocytes to come and chew it up and direct it for breakdown. So yeah, now we come to question number 12, where we have been asked about sphingomyelin. So this was already there in the direct questions. So sphingomyelin are most abundantly found in connective tissues, bone marrow, lung epithelial cells, or nerve cells. So the answer is, of course, nerve cells or neurons, where we know that the myelin sheath, which is like a coating which is present along the entire length of a neuron, along the axon particularly, is made up of, among other things, it is made up of sphingomyelin, and that's actually how the name myelin sheath comes. So there are some other cells also, oligodendrocytes, etc., which are present, but mainly the myelin sheath is there, and it has an immense role to play. So first of all, for normal, under normal circumstances, the conduction of signals along a neuron. So for in the neural network, because cells have to contact or connect with each other or relay information from one end to another, there is saltatory conduction, which is saltatory means jumping. So the myelin sheath is not continuous. If you consider this to be the length, this is the myelin sheath, again myelin sheath, again myelin sheath. So there are areas where it is discontinuous. And that is how the signal jumps and it travels along the length, thereby making it a much quicker process. So we are actually seeing that sphingomyelin is most abundantly found in nerve cells, and it has a huge role to play in nerve conduction as well. So just a second. Sorry. So nerve cells are and yeah, in fact the absence or the, I won't say absence, the degeneration of sphingomyelin, or rather the myelin sheath in certain regions, is actually what leads to several neurodegenerative disorders. For example, Parkinson's, that's one of the major disadvantages that we see. There's also, sorry, not Parkinson's, it's multiple sclerosis that is a result of myelin sheath degradation. So we see that there. Then so sorry, I keep getting one call. Just one. Yeah. So coming back to the next question, where we were asked which phospholipid is actually used as a surfactant. So this was also mentioned under the function of phospholipids. So dipalmitoyl lecithin is the answer to this. There's not much to actually explain about it, but yeah, it's a phospholipid that is commonly used as a surfactant as well. Now we come to this very interesting, very unfortunate, but very interesting disease, which is where we have been given the symptoms. So mental retardation, blindness, muscular weakness, and accumulation of GM2 gangliosides are characteristics of A, Fabry's disease, metachromatic leukodystrophy, Tay-Sachs disease, or Parkinson's disease. So we have the answer here is Tay-Sachs disease. So if you look at the right in the diagram as well, you'll see that this is non-mutated versus gene mutation in the hexa gene, which is leading to, just a second. Yeah, which is leading to beta hexosaminidase A deficiency, which leads to GM2 ganglioside accumulation. Neurons end up swelling, and it leads to a cherry red spot in the macula that leads to narrowing of blood vessels. So that's what we see. Then come waxes, which are esters of fatty acids with high molecular weight. We are looking at tetrahydric alcohols, trihydric alcohols, dihydric alcohols, monohydric alcohols, and the answer to this is monohydric alcohols. So these are also like fairly direct questions, not really much to expand on them. Now, this actually covers up the assignment for that week. And what we will look at now is the are some sample problems. So just give me a second. So the amino acid which lacks the beta carbon. So I've given answers with it as well for ease of understanding. So we have alanine, proline, glycine, and valine. So if you look here, the only amino acid, it's obviously glycine. If you look at the structure, you will see that it does not contain a beta carbon. It only has the alpha, and it's connected. It's also called the only anomeric amino acid. So that's what we see here. Then comes the amino acid with a thioether linkage. So the answer to that is only methionine is an amino acid that has a ether linkage because it has an O group through which, it has an ether linkage basically. OCH3 is present. So methionine, histidine, lysine, and arginine, among them, methionine is the correct answer. Then we have the amino acids that are used in ketogenesis to generate acetyl coenzyme A during catabolism. Leucine and lysine are the two amino acids that are present. And then we have the type of amino acids which are present within the interior core of globular proteins as well as on the surface of the transmembrane region of integral membrane proteins. Let me just go through the question one more time. Amino acids that are present, if you consider a globular protein, in its interior core, these amino acids are present, but they're also present if you look at the transmembrane region of integral membrane proteins. That is within the area that is in contact with the rest of the lipid bilayer. Basically, both the environments are hydrophobic. If an amino acid is present at the core of a particular protein, of a globular protein, if there's an amino acid which is present right at the core, it means it is not in contact, or it won't be stable when it is in contact with the cytosol or with is the amino acid that has a non-polar hydrophobic side chain. So that is the answer to this question. Then coming to question number 5, this is a fairly direct question. In thin layer chromatography technique, the phase that retards the movement of the molecule during the interaction of the sample is A, the solvent phase, B, the stationary phase, C, the mobile phase, or D, the liquid phase. Here the answer is that it's the stationary phase, which can retard or impede the movement of the molecule when interacting with the sample. Fairly direct question. Coming to question number 6, the amino acid that gets iodinated in thyroglobulin. So thyroglobulin, thyroid gland, it's a hormone from the thyroid gland. The amino acid that undergoes the entire process of iodination, which is a modification of that amino acid. What would be the answer to that? We have proline, arginine, lysine, and tyrosine. So tyrosine is the answer to that. Also a direct question. Hopefully you've covered it by now. Then we have the structure that is, the question is asking about the structure that is solely stabilized by covalent bonds. And among the options, we have primary, secondary, tertiary, or quaternary structure. So basic or the first tier of protein structure. So you have an amino acid sequence. These amino acids are obviously connected by covalent bonds during the process of translation. And that is resulting in the formation of a peptide, a polypeptide, or peptide, whatever, right? So that is the primary structure. But from primary, when you go to higher order structures, such as secondary and tertiary and quaternary, all of which have been mentioned, there are other forces, other stabilizing forces which are required for the secondary structure. You have alpha helix and beta sheets, right? Or beta strands. They all are heavily dependent upon hydrophobic interactions among the residues, which results in the compact packaging and results in the three-dimensional structure which you ultimately see in the protein. So for such structures, there is a huge reliance, not just on this, or not just on hydrogen bonding, but when you go to higher order structure, there are other interactions such as Sol bridges, etc., ionic interactions, which are all resulting in the stabilization of the entity as a whole. So the answer to this would be just the primary structure gets solely stabilized by covalent bonds. Okay. Then we come to the major driving forces for protein folding. So the process of protein folding is something that remains a mystery, has been a mystery. It's being studied, and I think it will stay sort of an enigma for a relevant period of time. But one of the major driving forces for protein folding, if we look at it, we have ionic interactions, we have hydrophobic, electrostatic, and solvage interactions, right? The like from that amino acid to the higher order structures that you proceed towards, one of the major driving forces is actually the hydrophobic interactions that is deciding how the protein will be oriented, what will be its 3D structure, and effectively what will be its secondary, and then effectively its much more higher order structure. So these are the major drivers when we talk about protein folding as a process. um, then the reagent that is used in the cleavage of a polypeptide chain after aromatic amino acids. So the options that you see here, these are enzymes, right? What they do, you're looking at trypsin, chymotrypsin, cyanogen bromide. Urea is not an enzyme. Urea is just a chemical reagent. Hence, that's how the question has been framed as well. So when we talk about aromatic amino acids, then among these, only chymotrypsin is known to cleave after phenylalanine, tyrosine, and tryptophan, but the others do not work in that manner. Trypsin, for example, cleaves after lysine or arginine. So this is extremely specific because these enzymes or these reagents have a different way of operating, but enzymes in particular, they have these binding sites, active sites where certain residues are present, and only interactions with certain other specific residues can only result in their functional form. So for that reason, they maintain a huge specificity when it comes to where they will execute their cleavage activity. Therefore, for chymotrypsin, that is limited to aromatic amino acids, and only after phenylalanine, tyrosine, and tryptophan do we actually see that happening. So that's that. Then we come to the method that is used to determine the 3D structure of the target protein. We look at chromatography, mass spectrometry, NMR spectrometry, and infrared spectroscopy. So it's obviously a well-known and extremely well-studied structure, extremely well-studied experimental technique as well, and that is NMR spectroscopy, right? Nuclear magnetic resonance spectroscopy. It's actually used to study study proteins in their native environment, in some sort of aqueous solvent as well. So that's what it is used for. So that actually is a structure that gives us, that's actually a technique that gives us the 3D structure, but the other options, they're not actually used for the same. For example, if you talk about mass spectrometry, it's used to study protein, but in a different manner. For example, if you have a mixture of proteins, or if you have, say, a complex of proteins, if there are 10 proteins which form a complex, you can use, if you know one protein among them, you can use an antibody against that protein to pull the entire complex down by a process known as immunoprecipitation. Immuno, of course, is because there's an antibody involved. Precipitation is because it is precipitating or settling down the entire, or pulling down the entire complex based on that immunomolecule, which is based on that antibody. And that entire entity, that immunoprecipitated complex, can then be subjected to mass spec, which is a technique where each and every one of those proteins can be annotated, can be labeled, or can be identified. So you can actually identify all the interacting partners of your protein of interest using this technique, and therefore it's extremely useful. It's used for other studies as well. Just giving you one example. So that's mass spec, but it's not giving you the 3D structure of one target protein. That can be done using NMR. It has its limitations as well, but then every technique has its own pros and cons. It depends on what is the question that you're trying to address, that you will decide which approach you should go about. Then comes question number 11, whereby the factor which does not modulate the binding of oxygen to hemoglobin to produce oxyhemoglobin in lungs is A, relatively high pH, B, relatively low H plus, low 2,3-bisphosphoglycerate, or D, increasing the temperature. We are talking about the binding of oxygen to hemoglobin to produce oxyhemoglobin. So temperature has no role to play here, right? pH. So when we talk about all of the factors that affect this entire exchange of oxygen binding and its release, there is pH, or H plus ion concentration, which is also what the second option is relating to. The third is BPG, 2,3 BPG, which also has a huge role to play. But temperature, of course, does not have it. You can study this via Bohr effect as well, right? So that's conclusive for question 11. Then coming to question number 12, where we are talking about the GTPase protein that is involved in the pinching of the clathrin-coated bits during LDL cholesterol uptake. So GTPase protein, there are RAN GTPase, Rho GTPase, RAP, etc., lots of them. But this pinching, particularly of the vesicles that are coated with clathrin, that is very well regulated and very finely controlled by the protein known as dandamin. Again, a direct question. It's actually a memory-based question. It's very, very detailed, but then yes, it's how you learn new things. Then coming to question 13. So pathogen cells and unwanted cell debris. Oh, sorry. Pathogen cells and unwanted cell debris is scavenged or removed from the body through a process known as exocytosis, endocytosis, pinocytosis, or phagocytosis. Now, from here, we have already discussed this in our previous class as well, previous interaction as well, that endocytosis is the process of taking something into the cell, right? Pinocytosis is making it more specific for liquids, but phago and exocytosis are different. So phagocytosis and exocytosis, technically you can say that it's doing similar function, but no. Exocytosis is the process of simply removing, or not removing, simply sending something out of the cell, or it is sending something to the plasma membrane, sending something to the cell exterior, to another cell. That's how it works. But when you are removing something which is not desired, when you are chewing it up, and that is being done by some specific cells that are commonly, you can call them phagocytes, or you can it's done by some specific entities which are trying to protect the cell, and therefore they are removing these entities. So under those circumstances, the process will specifically be known as phagocytosis. Phago is kind of like eating it up or chewing it up. That is the defining differentiation between these two. Then in question 15, we have been asked that during injury or wound, platelets are able to release a mitogen. Mitogen is basically any chemical or entity that allows a cell to undergo mitosis or divide. So a mitogen known as A, lipopolysaccharides, which is LPS, B, PDGF for platelet-derived growth factors, C, concanavalin A, or D, phytohemagglutinin. So here the answer is PDGF. So if not via any other option, you can see that platelets, if they're releasing a particular, the name in itself is quite direct. So in biology, you will come across a lot of instances like this, where platelet-derived growth factors literally means that platelets are releasing some factors, some components or entities that are allowing a cell to grow. When you're saying grow, it means eventually for the cell to divide, right? So they're releasing that particular mitogen, and it is PDGF, which is the answer to this question. Then coming to question 16, sample problem. We are talking about the molecular technique, which is used to identify the antigen in a biological fluid. So when you're talking about antigen, if you want to identify an antigen, you most definitely need an antibody. So if you look at the options here, the only immunotechnique per se here is ELISA. That's the easy shorter way of actually getting to the answer to this question. But logically speaking, southern blotting is done for DNA molecules, right? To separate and study DNA or nucleic acids. Not nucleic acids, I'll say it's for studying DNA. But northern blotting is for RNA, Western is for protein. Chromatography is a separation technique. So if your question is asking for one particular antigen in a biological fluid. ELISA tests are done so commonly. For example, for dengue, right? For dengue, the detection is done using ELISA only. But yeah, of course, for research purposes, it is very extensively used. So if you have that biological fluid, for example, if you have blood and you're trying to study something from it, whether this particular protein or this particular antigen is present or not, you will have a surface, you will localize antibodies on that surface, and then you will have whatever antigen binding to it, and you can find the solution, or you can find answers to your question. So that's ELISA. Then coming to question 17, the purpose of the blocking solution. So there's often a tendency to take blocking solution quite lightly, that why at all do you need to block a membrane? But it's a crucial step. It's crucial because because you will infer your results based on what is bound, right? Now that binding might be non-specific. So the entire purpose of blocking is to remove or to prevent any non-specific binding. And in case of ELISA, what it does is it is blocking the remaining protein bonding sites wherever the antigen has not already bound or the antibody has not bound, depending on which ELISA technique you're talking about. The remaining sites will be occupied by that protein. For example, if you're blocking using, say, skimmed milk, or you're blocking using BS, bovine serum albumin. Majorly skimmed milk is used because of its availability, because it's more friendly, animal friendly, more easily accessible, less expensive, lots of contributing factors. But what it does is it's blocking all of those remaining sites and it's allowing the cell to survive. So that's what we see here. And in question 18, which of the following is not correct regarding thin layer chromatography? So it's based on the adsorption. We talked about thin layer chromatography a while back, and we will discuss it next week in detail as well. But the questions here are asking whether it is based on adsorption mechanism, whether it depends upon the functional group which is present on the analyte, whether retention factor, RF, is unaffected by temperature and layer thickness, or whether polar compounds will have low retention values than non-polar ones under same developing conditions. Here, option C is the correct factor, is the incorrect factor, because we've been asked that which of these is not true. And in fact, yes, retention factor it gets affected by temperature, layer thickness. So that's the answer to this question. Coming to the final two questions for this week. So the amino acid side chain that is most vulnerable to oxidative damage is A, glycine, B, lysine, C, phenylalanine, or D, proline. The answer for this is lysine. Its extended side chain of this particular amino acid is often very, very vulnerable to oxidative damage, and also to other modifications as well. But here we are just restricting ourselves to oxidative damage based on the question that has been presented. So that is lysine. And for the final question, we have the type of the saturation curve that is exhibited in hemoglobin. You already know the answer while you're reading the question, but yes, for hemoglobin, when you look at oxygen and CO2 binding, the type of saturation curve, is it A, hyperbolic, B, sinusoidal, C, sigmoidal, or D, linear? The answer to that is, of course, option C, that is sigmoidal. So with that, we have discussed some sample problems. So just to brush everything up and wrap up the session for today, first, we talked about week 4, the entire content of week 4. We talked about all of the different biomolecules. We started with nucleic acids, its structure, its properties, what are its functions, the basic rules that actually help to identify how nucleic acids are made, how major is in a huge chunk of living organisms. They have DNA as the genetic material, and RNA is majorly restricted only to viruses. Then the structures, then Chargaff's rules, how A, T, G, and C are found to exhibit complementarity with respect to each other, and so many other things about it. Then we moved on to extensive discussion about carbohydrates, all the different forms of the same. We started with enantiomers, anomers, epimers, all of these different isomers. We studied them, we studied their function, how are they relevant. Then we eventually moved towards the entire metabolic not entire, but quite some depth about the metabolic processes in which carbohydrates play a huge role, starting with glycolysis, all of its detailed steps, what are the enzymes involved, what are the individual steps that ultimately lead to generation of that small eight molecules of ATP. Then we shifted towards Krebs cycle, a much more advanced, complex, but very, very relevant process. The huge number of intermediates, their individual functions, the relevance of each of these steps. We discussed how you need a mnemonic, or you can opt for a mnemonic to try and recall as much as is possible. And then we shifted from those discussions towards after Krebs cycle, we discussed a little bit more about its, a lot of questions about it, a little bit more about carbohydrates and their relevance, and how it ultimately generates 30 molecules of ADP from one molecule of glucose, and how that is controlled, how it is regulated by insulin and by covalent modification, and also by some other processes, like the allosteric modification of some enzymes. So all of that all together, it's the entire process is actually regulated. So that's what we understand over here. Then, then we moved on to lipids, and we discussed quite in depth about what are lipids, what are they made up of, how heterogeneous they are, the different subclassifications, glycolipids, phingolipids, phospholipids. We think about phospholipid, we immediately think about a bilayer, but it's not restricted there, right? It has other applications. It can have clinical relevance, like for example, think about vasodilators like nitroglycerin. You think about surfactants, etc., and all of their very, very diverse modes of actions or relevance. So we discussed all of that as well. Then we talked about the different classes, like how there can be simple lipids, there can be compound lipids, there can be some modified lipids as well, and how each of them has their own function to perform within the living system, and how they are each relevant in their own way. And after that, after we revised everything, we discussed the assignment, and finally we looked at some sample problems from the topics that are scheduled for the next session. We will of course start with a revision of the topics that have been discussed for week 5, just to sum it up, brush it up before we delve into the questions of the assignment, and for the upcoming week as well. So we will see how that goes. We'll learn about all of the individual processes in quite some depth, and it should all come together. So with that, I would like to conclude my session. I hope this is useful, and we will again meet next Monday from 7 to 9 p.m. and we will discuss about these topics for week 5 in quite some depth. And good luck with your assignment, and hopefully this course has been quite helpful to you. So thank you so much. Good night.